Hey, welcome back for the second portion of our lymphatic and immune system. So we talked about the non-specific defenses in our we talked about the non-specific defenses in our last topic. We're going to look at now the specific defenses. So what are things that are designed in our body to protect us, not a coincidence of another body system. So these take a little while to get going. They're high energy systems, so it's going to take us a couple of days up to a week before they actually activate. But once they activate, they're hopefully going to protect us and give us some immunity from various different illnesses. This is something that is mediated by antibodies, and there's two different types of cells that we're going to talk about. B cells and how they connect to B cell receptors. They're going to clone themselves, produce plasma cells, which produce antibodies, or they're going to produce memory cells, which will remember any illness for later exposures so that we have this immunity in our bodies. Let's take a look at what this is all about. First off, we have what's called the clonal selection theory. This is where you have an antigen that's going to bind onto a BCR, a B cell receptor, of either the B cell or a TCR, T cell receptor of a T cell. This causes that cell to clone itself. In production of that, you're going to get a couple of different kinds of cells. Plasma cells, which are good. That gives us a little bit of a protection on site, or it's going to create a few memory cells as well. And these are going to remember the illness for later exposure, as previously mentioned. At the very end of your illness, those cells, the plasma cells, are going to undergo apoptosis to destroy themselves, flushing them out of your system. The good news is that your memory cells are going to stay. They're going to stick around so that next time you do get exposed, you have that illness. You're prepared for that illness. So here you can see in the image, you got your B cells, the BCRs, when they're activated by the antigen, as you can see in the second image, which is also helped out by cytokines produced by your T cell system. You end up getting two different kinds of cells. Memory cells, which hang around, they still have the antibodies attached onto them, so they know how to reproduce. Or they're going to produce plasma cells, and the plasma cells are the ones that are going to eat up the pathogen and hopefully destroy it for you. At the end of that, you end up with apoptosis, where the cells will lyse themselves, and then we reuse what we can. Anything else gets flushed through our system. In a specific defense, we have these antibodies, which are going to try and bind onto some sort of antigen. Antibodies are this Y-shaped molecule. It's an immunoprotein. It's got two arms, a left side and a right side, and then it's got a couple of regions to the structure itself. A constant region, which never really changes. A variable region near the ends of the arms, which allows a little bit of flexibility for an induced fit model. And then lastly, you have a hypervariable region at the very end, which gives you really good variability to wrap around and induce a fit to an antigen. So it helps with the lock and key method, but I'd say it goes more towards that induced fit that we talked about a long time ago. When you have an antigen and an antibody combined, we say that we have formed a complex. So here you can see the antibody itself, and you can see the antigen that is bound to it, and then just a 3D rendering of what that would be as well. A couple of different kinds of antibodies. You can pause and read here if you want to take a look at the IgGs, IgMs, IgAs, IgDs, and IgEs, just on the various parts of the body where you're going to find these various antibodies. Now, after your B cells, you have your T cell network, which means you have to have a TCR, which is your T cell receptor. The weird thing about these is they don't actually recognize the antigen. They need some sort of assistance to do that. So we have what's called an antigen presenting cell, and it could be anything that can capture the antigen. So it could be some sort of phagocyte, quite often a dendritic or a macrophage. It could just be a phagocyte that destroys the pathogen and keeps a portion of it, and then brings that portion back to show the T cell receptor. When this is done, regardless of what kind of cell you have, we have this presenting cell and this whole complex together called the major histocompatibility complex. You'll find this on the surface of any antigen presenting cell, so any APC. And they're going to present it to the lymphocyte. We'll see what an image of that looks like in just a second here. When the T cell does end up getting a bond to this MHC, you end up getting this T cell activation, and that's going to get the clonal expansion, producing either cytotoxic T cells, which are going to help with the immunity and destroying of the pathogen, or just like the B cells, will produce helper T cells and memory cells. The memory cells are going to hang around for a bit. The helper T cells are going to activate the rest of the immune system. So they're going to help activate the B cell network so that we get both the T and the B cells working with us. So here you can see T cells, and you get the T cell receptors. They can't find the antigen on their own, so instead, a macrophage or some sort of phagocyte is going to hold on to the antigen. Together, they produce the MHC, the major histocompatibility complex. They make that complex together, and then that produces this clonal expansion for cytotoxic, which are going to help us fighting off infected cells or memory cells, which are going to remember the illness for later exposure. But we have those two types, cytotoxic. These are going to help us out. The way they work is they have inside of them both perforins and granzymes. Perforins are going to lodge onto the virus or the infected cell, and they're going to pry it open. So there's a teeny tiny hole to fit into. And then we have the granzymes, which are large enzymes. They're going to fit through that hole and kind of mission impossible it. They go into the infected cell and they destroy it from the inside out. The helper cells are going to secrete cytokines, which are going to help activate the rest of the immune system. As you can imagine, though, if this is the only cell that activates the immune system, there are illnesses where if you attack the helper T cell network, you shut off the entire immune system network. So that's what we get with the HIV virus, which we'll talk about in part 3 a little bit more. So here you can see a little bit of what's going on here. You've got your T cell, your cytotoxic T cell binding onto this virus infected cell. If we zoom in on that connection, you can see perforins are lodging in and making a small hole, and then the granzymes are entering. And here you can see how the T cells are doing so with a cancer cell, just as an example. Now, if we know how our immune system is going to work in these ways, then we can also talk about various types of immunity that we get. Induced immunity is when we put the immunity into a system, or you can have something that is naturally occurring. Naturally is always better, but sometimes we need to induce some sort of immunity, either actively, where your body produces its own immune network, or passively, where we produce antibodies that we can pump into your system. So, active immunity can come from immunizations, things like vaccines. When you get a vaccine, it accelerates antibody growth, and then you get booster responses, booster shots to really activate this immunity network. So I'll show you a graph of what that looks like. You get this first exposure to the vaccine, plasma antibodies increase, but then they start to tail off because your illness is gone. Second exposure, you get a big spike. And if you have a third exposure, you get an even bigger spike. So you get a much better response to vaccinations. Here's a list of a couple other vaccinations that you might have already had as well when you were younger. So just to compare those there. Now with passive immunity, this one's slightly different. We're giving you antibodies, so your body's not going to learn to produce them on its own, which means you only get temporary immunity. It's not going to last very long. You don't get any memory cells from this type of illness. So, as a result, we can prevent illness or we can protect you from certain illness, but it's not going to last very long. Okay, so a couple of ways that you can see that we can give you antibodies if you have some sort of illness. You might get some IgEs across the placenta if you are a fetus in the womb, or infants that are breastfeeding also get it from their mother's milk because their immune systems are not developed enough to produce their own antibodies yet. Now, probably one of the cooler parts of induced immunity are the monoclonal antibodies. These are a group of cells where you can create what's called a hybridoma. It's a Trojan horse of an immune system where we can take some sort of illness, fuse it together with a healthy plasma cell, and then reinsert that plasma cell into this unhealthy network so we can attack the illness from the inside out. We can also use it for diagnostic tests to see if certain proteins are there or not. This allows us to keep the antibodies alive a lot longer than they would be otherwise. So one of the more common ways of dealing with this is in a situation where you have a plasma cell which is infected with some other sort of cell. So in this case, a cancerous cell. It produces hybridoma, which contains the antibodies. They're going to last long enough to fight off the illness. We can insert the hybridoma back into a cancerous tumor, and then hopefully we can start to break it down from the inside and out. And that is going to bring us to the end of part 2, a very short session on some various types of ways our antibody network is going to work. In our next part, we'll look at a couple of illnesses and what kind of goes wrong with your body systems when the immune system starts to shut off.